Hello there, everyone. Thank you all for watching. I'm Kaiya Provost, and today I have the pleasure to talk to you about our work in avian bioacoustics. Birds show an astonishing array of songs and calls, and the diversity and functions of those have been studied across many facets of biology. This includes how song develops in the brain, how song mediates inter and intraspecific behavior, and how song contributes to the development of reproductive isolation, and thereby diversification and speciation. Most of our knowledge on birdsong originates from single taxon perspectives, from within species up to within families. Now, what I'm really interested in, though, is what is driving song differentiation, not just at those scales, but at the community level. The literature gives three main hypotheses for what drives song differentiation: the environment, adaptation, and stochasticity. So here's just a few examples of these. For reinforcement, Andean wrens show character displacement between two elevationally parapatric subspecies. For adaptation, we see that song changes in response to the environment. One example is the urban-rural gradient in white-crowned sparrows, and another is in the furnariids. We see that vegetation in the habitat, as well as beak morphology, constrains sound production. And for stochasticity, there are lots of examples of birds showing the development of dialects across space due to cultural drift, for instance, here in the Amazon parrots. Now, under each of these hypotheses, we can generate some predictions at the community level. First, we can see what other factors correlate with aspects of songs. So under reinforcement, we would expect songs to be correlated with genetic distance between communities, as well as population structure. Under adaptation, we would expect songs to be correlated with environmental or morphological differentiation. And lastly, with stochasticity, we would expect songs to be correlated with geographic distance between communities. We can also make predictions about how similar songs are within the community, so looking at interactions between taxa. With reinforcement, songs should be more different than expected due to character displacement. With adaptation, the same selective pressures, whether they're environmental or morphological, should cause songs to converge to be more similar than expected. And under stochasticity, songs should follow a null model of similarity, so be as similar as expected. Now, we've started looking at these questions by seeing how three of these processes impact the different aspects of song. Now, to get at the songs, we're leveraging the resources of multiple bioacoustics databases, and these vary in size and scope. So for instance, the Borer Lab of Bioacoustics here at The Ohio State University, where I'm based, has on the order of 34,000 recordings, and they're almost entirely exclusive to North America. Most of them were recorded in the 40s through the 90s. On the other hand, better known databases like Xena-Canto and the Macaulay Library are larger, with hundreds of thousands to millions of recordings, and they have a lot more modern data. Today, our data come from these first two sources, and in particular, we're focusing on one species, the white-crowned sparrow, which has been heavily studied with respect to its song. Now, given the magnitude of these data, we wanted to use a relatively automated process to summarize the sounds. And our first step is to develop a way to extract the syllables of the song. Fortunately for us, there's a software package in preprint right now called TweetyNet, which is specifically designed to extract syllables via supervised machine learning, specifically using convolutional and recurrent neural networks. Our setup has three basic steps. First, we annotate training data using Raven, which are these red boxes in this white-crowned sparrow song. Then we train TweetyNet using those data. And finally, we ask the trained model to predict where the syllables are in the dashed blue boxes. Once the syllables are identified, we use a recent R package called SoundShape, which is able to take those syllables and apply geometric morphometric principles to summarize them, specifically into a principal components analysis. Now let's look at a toy example to explain the metrics we'll be extracting for this talk. On the x-axis, we have principal component 1, y-axis principal component 2. These gray dots are individual syllables, and the colored ovals show different taxa. We extract two metrics from these data. One is song location, which is where that centroid point is, indicating by the colored crosses here. And this is a proxy for what kinds of syllables the taxa are using. In this case, the red and green ovals on the left have similar song location, whereas the blue one does not. And then we extract song diversity by calculating the volume in each of these shapes. So here, the red and blue ovals have the same song diversity, whereas the green has a reduced song diversity. Now, here are what these data look like for each of the 5 subspecies of the white-crowned sparrow. The subspecies here are indicated by color, polygons representing the volume across the subspecies, and the centroid points are marked with crosses. Note there are a few songs that we don't have subspecific data for. When comparing song location, Pugetensis in purple and Natalii in green both are statistically significantly different from each other and from the rest of the taxa, which suggests that there's some differentiation going on. This is not a novel finding. These 2 subspecies form a contact zone along the western coast of North America, and they're known to discriminate between each other's songs. But this does offer support more broadly for our reinforcement hypothesis, and it's nice to know that our automated methods are picking up on the actual biology of these organisms. Now let's look at song diversity, or the relative volume of those polygons I just showed you. Here are those data as a bar chart, with subspecies by those same colors on the x-axis, and the song diversity occupied by each of them on the y-axis. And this diversity is standardized by the total song diversity occupied across the whole species. Individual subspecies occupy roughly 80% down to 40% of the total volume, with the nominate in dark blue having nearly twice as much volume as Pugetensis in purple. And notably, these 2 differentiated subspecies, Natalii and Pugetensis, have a much reduced volume compared to the others. And this could be due to character displacement causing these species to use different parts of their vocal repertoire. We can also look at these data not from a subspecies standpoint, but rather from a geographic standpoint. So here on the right is a map of the song diversity occupied in each grid cell across North America, with warm colors showing high values, cool colors low values, and gray showing no songs present. You can see that the song diversity across the species is very uneven, but likewise that the sampling across the range of the sample so far is uneven, which we're going to have to account for. And just for context, this arrow shows the location of the Natalii Pugetensis contact zone I mentioned, which has received a lot of attention and has a lot of sampling. Okay, now with those data, let's look at the adaptation hypothesis. Specifically, we're going to examine the impact of environment on song diversity and song location for each of those grid cells. We use 2 layers from WorldClim, specifically mean annual temperature and precipitation, and we're evaluating whether environmental factors can be playing a role in differentiation. Here we're using these climate layers as proxies for habitat, which is known to impact songs across many species of birds. I'm showing you here results from one of our analyses. So on the x-axis is the difference in temperature between grid cells. The y-axis is the difference in song location. We find a significant positive correlation here with an R² value of 3%. So areas with highly different temperatures also show differences in which syllables are being used. And this does hold after accounting for sample size. We don't find any relationship between song location and precipitation though. And on top of that, there aren't any differences between the relative song diversity in any of these regions. Sample size bias is a much better predictor of relative song diversity than temperature. So our conclusion here is that habitat changes can cause the exact syllables that are used to change, but it doesn't cause differences in the overall variety within songs, at least at the species level in the white-crowned sparrow. Now, with those same data, we evaluated the stochasticity hypothesis, specifically the influence of geographic distance. And again, we find a significant correlation between song location and geographic distance with an R² of 10%. So the exact syllables that are used do change across space, so there are dialects, and this holds even after you account for sample size. But this is not the case, again, for song diversity, which has a non-significant relationship with geographic distance and is, again, entirely explained by the sample size in the grid cell. Okay, today I've talked about 3 potential hypotheses that describe song differentiation in birds. And I focused on one species as we've developed our automated machine learning and our geometric morphometric methodology in preparation to expand. In the white-crowned sparrow, we find some support for the reinforcement hypothesis. So the Pugetensis and Natalii subspecies do show differentiation in their songs, both in terms of differences in syllables and an apparent reduction in the song space used relative to the other taxa. When we look at adaptation, the different syllables used are correlated with the environment, especially temperature. This may reflect differences in habitat that are constraining the types of syllables and songs that are used. And lastly, we do see that the sparrows have different dialects that are used across geographic space, which supports the stochasticity hypothesis. So in this single species, we find some support for all 3 hypotheses, but we're currently expanding our sampling to encompass many more taxa, of which just a smattering is here. With a lot of species distributed across North America, we will be able to investigate the relative importance of these hypotheses across the community. Finally, we're also starting to add more sources of information on these species, with our first priority being genetic information. The lab I'm currently in, the Karstens lab, is developing Phylogator, which is a tool that queries GenBank as well as other databases like GBIF and automatically produces aligned georeference sequences for any taxon. So this, plus our plans to add in more detailed environmental and morphological data, will give us a really fine scale resolution of the biogeographic history for all of our study groups. And with that, I have a lot of people and institutions to thank, and I would be happy to take questions. Thank you.